Oi, Corado.
Boa tarde, professor.
Oi, posso deixar deixar a pessoa entrar, que ainda eu não preparei. Está preparada a moça?
Pode deixar que eu vou botando, sim.
Cássia, fica muito querido. Oi, Rodrigo.
Olá.
Vou deixar um segundo você, que eu ainda estou com almoço, não consegui almoçar.
Ok.
5 minutos, deixa você entrar.
Hi,
Olá, Jama, ¿qué tal?
how are you?
Bem, e tu? Como estás?
Bien, bien. Hi, Stefano.
Thank you very much for the reference.
So I've been thinking also about papers for your students, and so today I'm gonna comment on some papers that might be interesting to look at, and also I want to look at. But I wonder if you were interested more in classical papers in the field or more like recent results.
I will try to get that that seminar that I listened to when I was in Belgium. And I will write you about the idea. It was a sort of very simple, I think it's easy, a simpler switch. Is that with living with parents, they had some of this cardiovascular problem, then people got more observant about some mechanism and difference in human beings. So
I see.
Because, for example, the difference between Alzheimer and the usual cardiovascular program that are not Alzheimer, but they affect more the hippocampus. So that kind of difference in the modeling is very interesting, because I've been told the Alzheimer is more affecting the pathways of the neural networks, while instead the hippocampus is more affecting the group of cells, the neural, how you say, the cluster of neurons that did not receiving oxygen because of the lack of the capillary got obstructed. So it's how this can be reflected in the modeling, this kind of because one and this at the level of exams, I know that now there is a more sophisticated one, but in the usual exam, one is the tomography, the other is magnetic resonance, and they put in evidence different kind of behavior, they're kind of complementary. And so how this kind of information, because reflects in the model, like, because, for example, the tomography is more like if the pathway are free, and instead the magnetic resonance, if this response, I want to do something, can I, so the decision making that you were mentioning. But I would just like to learn, because I'm just been talking to neurologists that they've been telling me about these differences, but I don't know much. So I'm really like in a primordial, in the crash, as you say, in the maternal school. So, but I think it's very interesting, because I see that some of the doctor, if you don't speak with specialists themselves, they confuse the two things. And they're crucial difference, because in the cardiovascular case, you can do something. You can do more exercise to create more capillary to oxygen. So to kind of stop this reduction of the hippocampus that is basically is lack of oxygen. While instead, if there is a pathway, is like kind of the real Alzheimer. I don't know if now there are more, from time to time we hear that they understand how to take away this barrier to the pathway, but it's a totally different problem, even from the point of view of the treatment. There is no treatment. In the other one, you can act, and it will be good to have some models, that could put in evidence this different kind of structure. And it's so at that time, at that conference, and I heard in Belgium, that was in evidence of a kind of signal, long wave, short wave, this kind of response was, it could be mapped into some of these signals. So I have to find again this, and I will send you, because that kind of trigger, more than intuition that, I don't know, but I thought it was very interesting, because after when I talked to doctors, they seems, some of them, for example, you have some doctors that will only prescribe the tomography and not the magnetic resonance, but you can have a clean tomography, no Alzheimer, but you can have a problem in the hippocampus. So the two are, and so, so, okay, but, I will send you, I will try to get back. This is a group in the Netherlands, nearby, it's not Amsterdam, it's close to Amsterdam. And I thought that it's one of the first group in Europe that understood that it has to be done a very individual treatment for this kind of disease, because you have to map how is your situation. You can standardize. And so already at that time, they already, I'm talking about.
You're muted.
Already 4 or 5 years ago, they were applying this treatment to even to Parkinson people. And apparently this student was a student that was finishing her PhD. She had her grandmother. Since she was part of this group, so her grandmother was part of, and she had very positive results. I mean, they could connect, you know, part of the brain that were disconnected. They could. I know that even here in our university, they have a sort of capacete, a sort of helmet, where they have some sensor. They can connect different parts of the brain, but I don't know. I talked once to the guy, and I don't know now how they progress. So all this is very interesting. How things, you know, to characterize this different kind of neuron modeling, you know, and especially like to understand the hippocampus because it seems. But okay, but we can talk some other time. So, but it's very. I will send you back. I will try to get back the name of this the paper.
Yeah.
Because I remember.
Do they do? So they are. Do they do modeling or they just do experiments?
Yeah. No, they were doing modeling, but at that time. They were doing. It's a group of mathematicians that was working with doctors. And they were analyzing the brain signals. I don't know. I don't remember now. I know that, for example, here in Rio, they have patients that they stay there and they connect for some time. For example, we have some people, Dr. Edouard Faderet, very good. He worked at the Hospital of the Brain, and he has a huge collection of data from his patient, but he's more concerned about epilepsy. So this is why also I ask you about epilepsy. And he's looking for people doing modeling on this huge set of data. And so will be very interesting. So but so but these are patients that sometimes are there in observation, you know, have this needle, and this of the this helmet is less invasive because it doesn't go deep. You see, it's more. So it's a different, but and it can have sensor in different part, you know, like So. Yeah, but I will try to send you the one of Belgium, because now I know that in France, years later in France, now they have the spin, I think the spin in the Saclay Plateau project, where now they also run after the fact that has to be an individual treatment, and but the Dutch were first, were ahead of everyone, you know. So I always say to the students, you know, it would be good if you're interested in this, look this group, at least from if you want to do something practical, you know. I think it's the Delft group, University. UT Delft. But I will send you the very interesting work.
Okay. Yeah. Good.
Okay, great. Yeah, it's fascinating, and then as we get older, we know that this is very relevant even to us. And when you talk to doctor, I mean, I discovered that some neurologists, there are specialists, you know, even between the neurologists, so you have the general neurologist, and then you have the specialist at camp. And it was fascinating to talk to neurologists of hippocamp. I found that the one of hippocamp, they are so much more knowledgeable. I don't know what kind of study they do, but they have such
Yeah.
such a vision that, you know, that explained to me that people are affected. One of our colleagues, they explained to me that they all call dementia, but actually you have the one that is only Alzheimer, or the one they only have cardiovascular problem, or some people are very unlucky and they have both. So now we heard about cardiovascular problem because we heard about COVID, but it's always present there.
No, it's one of the biggest causes of death, in the world.
Yeah, and it seems that some 20 minutes of aerobic exercise, it helps so much because you create a new capillary. And so this is how you can help your hippocamp to improve this oxygenation. So some
20 minutes per day.
Yes, 20 minutes per day is like a dose. You have to, they explained to me, it's not just walking. You have to go a little bit fast. So, you know, to be, and in fact, when I was in Spain, actually, I went to the gym and they have this elliptical machine. And then I experienced, I understand why it has to be 20 minutes, because if at the beginning, also I have to stress that I want to go to elliptical. At the beginning, I didn't have so much minutes. I have to train. But when you can go beyond 12 minutes, you see that after the time, your brain, you see it. It gets, you change state after 12 minutes. Your brain gets oxygenated, and when you get out of the machine, you feel better. It's something that you can feel it very concretely. So elliptical machine would be very. I mean, if they were
Very nice, very nice that you talk about this now, because I'm completely sedentary. I bought this bike. I don't know how to say.
Yeah,
but it's the bike. Because I'm not leaving the house and I'm not doing any exercise, so I bought this. But I was trying to do half an hour per day, but there are days that there is no time, and then not doing anything. But it's quite important. You're right.
Yeah, it really, I mean, it really changed it. But because I tested this 20 minutes, and it's really true. But you have to go to some pace. So actually, but you know, for example, here in Brazil, we don't have the same kind of quality machine that you can buy over there in Europe or in the States. You find in the gym, in the gymnasium, but not for, you know, for you to buy at home. And it's, you know.
Well, in general, we don't really need gym because
you go outside.
We can walk. We can run to take the subway. We can do stuff. For me, it's already exercise. I go up on the stairs running in the subway. You go to the university. It's already an exercise. You teach it. You finish a class. You finish a bit tired. It's some exercise, but without doing anything, all day sitting in the chair. Yeah.
Okay, great. So, so let's start for the last great lecture of this mini course. So I have to say, as Jacqueline said, even in the YouTube, students are really grateful to your lecture. There are a lot of, it's very inspiring. Even I'm giving ODE, so to see other, you know, this bicycle things very interesting, even for example in class. Okay, so let's start this last lecture. So please, Jama.
Okay, thank you, Stefanella. So we're gonna start with the last lecture, and today I want to show you an application of the models that we've seen in the previous sessions, or like some of the concepts that I introduced in the previous section. Let's see how they apply when we model. We want to model a real experiment. And in particular, I want to focus on the topic of decision-making and perception. Okay, so well, again, this is the outline of this course. So So when we talk about decision making, so this is going to be a disclaimer for this lecture, is that I'm going to present like so if you write decision making in Google, you're going to find like thousands and thousands and like a lot of material, a lot of studies, a lot of papers dealing with this topic, ranging from neuroscience to economics to many disciplines. So this is a very broad area of research. And today I'm just gonna give you like just a small taste of it. Okay, so I'm gonna simplify many things. So this is a disclaimer for today's class. So what is decision making? This is the process by which a sensory input is processed in the brain and then is transformed into a percept, and then an action is produced based on that. So a decision is taken. Okay, so how the brain does it? So of course, this is a very complex process, and depending on the decision we have to take, it's even more complex. So if we want to study it in a systematic way, we need to set like a sort of experiment that can be done in a lab and reproduced several times. And a classical experiment that is done in the field is called this two alternative forced choice task based on this stimulus called random dots. What is this stimulus? So a subject is presented with a visual stimulus where there appear several dots. And in some cases, like the ones here on my right, in some cases, we have that all the dots are moving in the same direction. And then we have to say in which direction, so the answer that we have to give is that in which direction the dots are moving. And typically we are given two options. So in this case, it's like downward or upward. So in the case that I have here on my right, this is an easy decision to take because it's very, it's simple once you see the stimulus to detect that the movement is downward. But in these experiments, what they do is that they change the number of dots that are moving in the same direction. So for instance, in the one in the middle, only 30% of the dots are moving in the same direction, in this case downward, while the other ones are moving in random directions. So when we have to take a decision about what is the motion here, whether downward or upward, this decision becomes more complicated. And it's even more complicated when all the dots are moving randomly. So there is 0% coherence, because in this case, our answer is going to be totally random. So to illustrate this task, let me show you a video where you're going to see these examples of this visual stimulus. So what we're going to see is this stimulus where we have dots moving in several directions, and we have to decide what is the direction of motion. In this case, whether it's rightward or leftward. And what we're going to see is that the percentage of dots that are moving coherently or are moving in the same direction is decreased over the video. So when we start, it's like 80% of the dots are moving coherently, then 60%, then 40%, then 20%. So, and let me show it again. Oops, sorry. Let me show this. Sorry. Where was I? Yeah. So you can see, now you can see, you would say right, but the decision is every time more difficult to take, especially when the coherence is only 20%. Okay. So this is the type of stimulus that is presented, and the subject is asked to decide between two options, either right or left or upward, downward. It depends on the stimulus. Okay. So this is a task that is done in experimental. Monkeys are trained to do this task. So they are sitting in front of a screen. First, there are no stimulus, and then at a certain, and they are given two options. Either they have to choose, they're gonna choose either right or left. These are the two options that are given. Then the stimulus is presented, and once they know, they take the decision, basically moving the eyes, okay? Either rightward or leftward, okay? In this case, the monkey is moving the eyes to the rightward direction. There is something that is also recorded, is like the reaction time, or how much time it takes the subject once the stimulus is presented to take the decision. Okay, of course, when it is more difficult, this reaction time increases. So this is a classical experiment by Rodman and Shadlen in 2002, and also there are similar ones in previous studies, and it's very classical, a classical experimental paradigm in the decision-making studies, experimental studies. So what do we want to know? We want to know what's happening in the brain when the subject is performing this task. And for this, basically, they are recording from two different areas of the brain. In this case, this is a macaque monkey. They are recording from the empty area in the visual cortex. So this is an area in the visual pathway that is at a high level. And in this area, there are neurons that are direction selective. This means that they have a preferred direction of motion, and they respond, they have activity when a visual stimulus is presented showing the movement that they like. On the other hand, they are recording also from this LIP area, the lateral intraparietal area, because there they have found decision-related neurons. What do we mean by decision-related in this experiment? So they have found neurons whose activity is correlated to the eye movement that the animal makes. So if the animal, there are neurons that when the animal moves the eyes to the right, they respond, while neurons that they respond if the animal moves the eyes to the left. Okay. So this is how they try to understand how decision is taken in the brain, or what is the process that is taking place in the brain when the subject is doing this task. So let me explain first a little bit about these direction selective neurons in the empty area. So this is a simplification, but in experiments, what they do is that they record from different neurons in the empty area. At the same time that a stimulus consisting of a certain movement in a certain direction is presented. And they try to establish the neuronal code. So for instance, when the upward movement is presented, so that the stimulus shows these dots moving all coherently upward. For instance, in this experiment, they observe the following spike train. They observe that, for instance, the first neuron is showing high activity, it's showing a high frequency of spikes, while the other ones, they show like just a few spikes or almost no activity. So we are going to say that the first neuron encodes the upward movement, And we're going to label it with this sign. Okay, so if we repeat now the experiment with, for instance, dots moving to the right, we observe what is the spike train of these neurons, and we observe that now neuron number 2 is the one that shows high activity or high firing rate, while the other ones, they show low activity. So we are going to label this neuron as having the preferred motion to the right. Similarly, we do this experiment with the downward motion. We observe what is the spike train, and we label the third neuron as having its preferred motion, as having the downward motion as its preferred one. And the same way for the leftward motion. We observe that this last neuron shows high activity, so we label this neuron according to this. What happens now if we're showing this movement like southwest? We look at the neuronal responses or the spike train of these neurons, and what we observe is that those neurons which are close to this those neurons with direction of motion, favorite or like preferred direction of motion that is close to this movement direction, they show some activity, okay, while the other ones show low activity. But notice that this activity is lower than when its preferred direction of motion was shown. So in general, for these neurons in the empty area, we can plot what it's called a tuning curve. So if in the x-axis we put the movement direction of the visual stimulus, and in the y-axis we put the firing rate response. So for the neuron that has its favorite or like its preferred direction of motion, the one downward, we're going to see that it achieves a maximum when the direction of motion is the downward one. And this, the firing rate shows a decay for nearby as we move away from this preferred direction of motion. And of course, for the opposite one, the activity is at its minimum. Okay, and this, we have tuning curves for all active at the same time. Yes, I'm going to show you now in a second. This is, for instance, this is what I shown you was a schematic representation, but this is, for instance, an example of an experimental recording made of a neuron in the empty area when several directions of motion are presented, okay? So as you can see, when this direction of motion is presented, the neuron shows high activity. While when we present the opposite one, the activity or the number of spikes per second is low. And for nearby directions of motion, so the activity shows certain activity even if it is lower than the preferred one. Okay. So this empty area of the brain contains like neurons that are direction selective. And by direction selective, I mean this, that they activate, that they have each neuron or a group of neurons. This is not a single neuron because actually nearby, so the empty area is structured in a way that nearby units have similar preferred directions. So you have groups of neurons that have a preferred direction of motion, and when it is presented, they show high maximum firing rate. So regarding the question, I think that with the next slides, I'm going to answer the question that is in the chat about like several neurons active. So suppose that now we want to read from the spiking activity of the neurons and decide and determine what is this type of stimulus, that visual stimulus that is presented. So if we see this type of spike trains of neural response, we would say that basically there is a stimulus moving leftward, because the one that has preferred direction leftward is showing high activity. And effectively, this is what happens. But in some cases, we have this situation here, for instance, is that, well, it seems that the one encoding the leftward motion is the one that is showing the highest activity. But these two other, they also show some activity, and even this one. So what we have in this case is that most of the dots are moving leftward, but some of them are moving in other directions. And in this case here, for instance, what we have is that it seems that all neurons have some activity. Even it is not as high as when as in those cases in which only one is firing. But in here, what happens is that we have that the dots are moving randomly in all directions, and all the neurons are responding, but with lower activity than when we have the coherent motion. Okay, so this is a little bit of a summary of what's happening in the empty neurons as a function of the motion coherence. That is how many dots are moving in the same direction. So you can see that when there is 100% coherence, in this case, the neuron with encoding for the upward movement is showing high activity. And as we reduce the coherence, the activity of the other ones is increasing while this one is decreasing. And in those cases where we have no coherence at all, then we have all the neurons like showing some activity, even if it is lower than in the 100% coherence case. Okay, so this is like, let's say, a simplification of how the visual stimulus showing movement in a certain direction is translated into a certain activity in this area MT. But now, based on that, we have to take a decision, because in this task, okay, going back to the decision-making task, out of this, we need to make a decision at some point. So remember that I told you about like two main regions from which the experimenters are recording, and the other one is the LIP region, in which they found neurons whose activity is correlated to the decision that the animal takes. So going back to our experiment, so the animal takes this decision, just moving the eyes, okay? And then what they do is that when the animal is doing this task, they record from LIP. And these are, for instance, the experimental recordings obtained from a neuron in the LIP region of the brain while the animal is doing this task. This neuron, the activity of this neuron is correlated with the decision that the animal takes, in the sense that when the animal decides right, okay, so moves the eyes to the right, this neuron shows high activity. While if the animal decides left, the neuron shows low activity. So what we have here, this is a trial in which the animal has decided right, okay? And what we have in this trial, this is a raster plot showing the spikes of the neuron for different trials for which the animal has decided right, okay? So each line corresponds to a trial, to the spiking activity of this neuron. The triangle here marks the moment at which the stimulus is presented, okay? The time at which the stimulus is presented. And the vertical line here indicates the time at which the animal makes the decision. So they have aligned all the trials so that the time at which it takes the decision is aligned. And what you can see here, when we look at the histogram of the activity, so just counting like at each time being how many spikes we have, this is the picture that we have here at the bottom, what we see is that this neuron is showing low activity. So here the stimulus is not present because the stimulus appears at this moment of time. And then once the stimulus appears, its activity starts to, the firing rate starts to goes up. It reaches its maximum once the decision is taken, once the animal moves the eyes. At this moment, the firing rate activity of this neuron is at its maximum. And once the decision is taken, it goes back to a low activity state. Notice what happens when the animal, this is the same neuron, but these are trials for which the animal has decided left. Okay, so when the animal has decided left, this neuron shows all the time low activity because there's another neuron whose activity is correlated with the decision left. And at this time, for these trials, sorry, what happens is that this other neuron shows high activity. Okay. And the difference between these raster plots and these ones here is that in one case, the stimulus has 50% coherence. At 50% coherence, like 50% of the dots moving coherently to the right, and it's easy to take this decision. So notice that the time that has elapsed since the motion is presented, since the stimulus is presented, until the decision is taken is kind of short compared to this other trial here where the coherence is lower, so the decision is much more difficult. So the animal takes more time, the reaction time here is much longer to take the decision than in the case where the coherence is 50%, because it is more difficult. So the animal takes more time to take the decision. And also, notice also that for this case, the activity of this neuron that likes the decision right is also ramping up until it reaches its maximum once the decision is taken, and then it decreases. Okay. Notice another effect here is that when the animal takes the other decision, the left one, the one that this neuron doesn't like or is not coding for, the activity is low. Is much lower that once the decision is taken, the activity also increases a little bit. So what's happening here, and I'm anticipating something that I'm going to explain later, is that once we build up, once the neuron is like increasing its firing rate to take this decision, what it does, it also inhibits, it sends inhibition to the other neurons coding for the opposite decision. Because you want to strengthen, so you want to have this neuron to have high activity and the other one low. So that once the decision is taken, this releases the other neuron, or like it stops inhibiting the other neuron, and this neuron recuperates certain activity, as it happens here. What is happening here, since the animal has taken the other option, is that there is another neuron that is suppressing this one. And once the decision is taken, this neuron is released and recuperates certain activity. Okay, so we're going to see this in a model. Let me also say that they also, in this experiment, the decision time be reduced if the trial is repeated several times. Actually, there is certain variability in the reaction time. So even for these cases that I showed you here, so this corresponds to several trials, and notice that the reaction time shows certain variability. So this one is a little bit shorter than this one. Or even here, you can observe this one is, in this trial, the animal was faster than in this trial. Of course, with this, so I am not an expert with this, but when you do this, when they do these experiments, the animals also have to have some resting times, because it can happen that at some point they get tired or they don't care about the decision. So you have to be careful with all these other aspects of the experiment, is that at some point you maybe just randomly reply or answer, or also if you, this involves also other aspects of the psychology, no? Not only for animals, but also for us. If you have several trials in which you have failed, then you are not as good in the next one, because you inherit like a sort of like a bad mood. And this also happened to the animals. So the experimenters have to be careful when they perform several trials to avoid biases due to the animal becoming tired, etc. That affect, of course, the reaction time. So these are recordings from neurons. These are like behavioral data from the experiments that they show, depending on the coherence of the dots, the percentage of dots that are moving coherently, what is the performance. And as you can see, is that around 20, when you have like between 20 and 40% of the dots that are moving coherently, then we're almost always correct. Okay, we always get what is the right direction of motion. And below we have this psychometric curve that the performance decreases until at 0, of course, it reaches like the 50%, because it's random, the decision, when it's 0%, because actually there is no correct answer. And also notice that the reaction time, when the task is harder, because there is no the coherence is 0, the reaction time is longer than when the task is easier, then the reaction time shortens. Okay. So let's talk about a model for this. Okay, so this is what we have from the experimental lab. But let's try to build a model about what's happening here. So I'm going to explain you a model by Wang in 2002, and then also a new version of the same idea by Wong and Wang in 2006. So these are maybe nice papers to look at if you are interested on that. There's a question. How does the time scale of the dots moving is chosen experimentally? The time scale of the dots moving is chosen experimentally. You mean the velocity? I'm not sure if I understand the question. How does the time scale of the velocity? I'm not sure. I'm not sure like how they choose the velocity. Yeah, you're right. So there are several parameters here that affect is that if things are moving faster or not, it might affect the performance. Yeah. The idea is that in all the experiments, you have to keep as many parameters as possible fixed, and then based on that, just modify one or like, but keep the others. But it's true because there are a lot of other parameters that might affect the performance. So going back to the model, so I want to tell you this model proposed by Wang. And remember what we talked in the previous lesson is that when we want to build a neural network, we need to decide like how many populations we're going to put, what type of cells we're going to have, inhibitory, excitatory, what do these populations represent, etc. So this is a spiking neural network model for which they have chosen two populations, the one indicated by the blue one and the red one, which are selective or like selective to the decision, okay, whose activity is correlated to the decision. Basically, population 1 activates when decision 1 is taken, and population 2 activates when the decision number 2 is taken, okay? Then there are these populations, they have self-excitation. So they send self-excitation to themselves. And also there are other populations in the in the neuronal cortex, that they are non-selective, that we have neurons that are there and maybe they are not selective to any of the decisions. On the other hand, these populations, they send excitation to a group of cells that are inhibitory, and they send back inhibition to this group, okay? This is a model with thousands of neurons that, and is a spiking network model in the sense that we model the voltage of each of the neurons in the cell, in the model, in the network, and we have an equation describing the dynamics of these neurons. And this, we end up with a system of like 7,000 equations. Of course, for this system, the only thing we can do is just simulate it in a computer and plot the raster plot. Remember, like for each neuron in the population, we are gonna plot, we're gonna put a dot every time this neuron spikes. So here in red, we have the neurons corresponding to this population 2, and in blue, we have the neurons corresponding to population 1. And we run one simulation. Sorry, before, let me say that I forgot to say is that these selective populations, they receive input, an external input, that we may think that is input coming from the empty cells, the empty cells that are direction selective, and that they respond according to the visual stimulus that they observe, okay? And they are sending input to these neurons that finally they're going to take the decision in the LIP region, okay? In the cases when there is 0% coherence, we might think that the input that these two populations receive is the same, in the sense that from the empty cells, we don't have any information that there is more dots of a certain with a certain movement than others. So we can think that they receive the same input. While if we have a higher coherence, maybe one of these two populations is going to have a higher input, because the input coming from the empty cells is going to be a little bit biased, in the sense that those neurons coding where most of the dots are moving are going to have higher activity than the other ones. Okay. So said this is that, so this is like sort of input from the empty cells. So if we simulate the system, in the case that we are injecting two equal inputs to both populations, so that there is no information from empty cells, in the sense that there are more dots moving in one direction than in the other. So in the case that the coherence is 0%, the spike, we run a simulation and we observe that in trial number 1, we have that the spiking activity of the population number 1 and the spiking activity of population number 2 is low and similar at the beginning, as you can see here in the average firing rate activity, low and similar. And then at a certain point, what happens is that the activity of population number 2 ramps up, while the activity of population number 1 goes down and is sort of suppressed by population number 2. And if here we have to take a decision, we are going to decide number 2, okay? Because populations coding for number 2 have higher activity than population coding for decision 1. In another trial, we can have that the simulation produces like the opposite result, in the sense that this is equal than in the previous one. At the beginning, the activity of both populations is low and very similar. But then at a certain point, what happens is that one of the populations, the activity of one of the populations ramps up and reaches a high value, while the other one is suppressed or kept suppressed, okay? So this is for the spiking model, but remember the previous class, what we said is that in the end, what we want is information about the overall firing rate of the network. We don't need the information, the detail of the information of like when or what are the spike times of each neuron in the model. We just need the overall firing activity. That is, we just need this type of dynamics to know. So 1 and so in this paper 1 and 1, they went from the spiking neuronal network model to a reduction based on this mean field approach. Okay, so you have this. You have the details of how they do this reduction, but in the end, it's just considering that a mean field approach describes the firing rate activity of each of these populations. So we get rid of all the neurons inside each population, and we just describe the firing rate activity of these populations. And then based on that, we can go from 7,000 equations to just 11 equations. This is already a very good simplification. But also using other hypotheses, we can make this simplification, we can bring this simplification to a higher, so we can simplify even more. And one of the strategies to get rid of the non-selective cells, because it seems that maybe they don't have an important role. And we can already reduce to 8 equations. And then we can make an extra simplification, which is get rid of these inhibitory neurons using the idea that the inhibition is going to be faster than the self-excitation of these neuronal populations. And then instead of, so since this is going to be fast, instead of thinking that we have our population that is activating an inhibitory population that at each time is suppressing back, we're going to assume that this is fast. This inhibition is direct, okay? And then we reduce to a two-dimensional model consisting of just 2 populations, population number 1 and population number 2, that they self-excite each other and that they send inhibition to the other one. Okay, and they receive an external input. Okay. Interestingly, they show in this paper that with the spiking neuronal network model and with this reduced 2-dimensional model, they can reproduce the same psychometric curves on observed in the experiments regarding the percentage of correct responses and the reaction time. Okay, as you can see here. So the graphics look pretty similar. Since this is a 2-dimensional model, so let me spend a little bit of time explaining the dynamics of this model. Okay, so let's perform a mathematical analysis of the model. This is not exactly the model that they derived in the paper. It's an ad hoc adaptation, but it's, let's say, it's the same idea and it works in the same way, okay? Although it's slightly different and involves other concepts that I would need more time to explain. How does this model look like? This looks like similar to the Wilson and Cohen equations that we saw the other day. So we have, this is a model with 2 variables, variable R1, which codes the firing rate of population 1, and variable R2 that describes the firing rate of population number 2. Okay. And how is the dynamics of this firing rate? Well, it has this expression here. So the rate of change of this firing rate, it is has this expression here. Again, this function S is one of these sigmoidal functions that go from 0 to 1. Okay, we might think that the activity the maximum activity of these populations is normalized to 1. Okay? So what we have is that once the total input that this population number 1 receives exceeds a certain threshold, this S is equal to 1, and the firing rate activity of population number 1 tends to this higher state value. While if the total input to this population is below the threshold, then S is 0 or is close to 0, and the firing rate activity of population number 1 is low or tends asymptotically to this low value. What is the total input to population number 1? Well, is the self-inhibition, which is represented by this term, and is proportional to the activity of the population, okay? With this factor W plus. So the more activity has population number 1, the more self-excitation sends to itself. What's the other input that this population receives? Well, it receives inhibition from population here with weight W minus. It receives inhibition from population number 2. So this goes with this negative sign. And of course, it's proportional, okay, to the activity of population number 2. The more active is population number 2, the more inhibition sends to population number 1. And finally, the external input, I sub 1, okay? The equation for population 2 is totally symmetric, is exactly the same, okay? So basically we have two populations that they self-excite and that they send inhibition to the other one. Since this is a two-dimensional model, we can do phase plane analysis, okay? So let's consider first the case where the external input is 0. This means that the animal is sitting in front of the screen, but the input is not presented yet. So the stimulus is not presented yet. And then we plot the null lines of this two-dimensional system. Okay, this S, you might think that S is R here, okay? So we plot the null lines of the two-dimensional system, of the two variables. And remember, the places where they intersect correspond to equilibrium points. In this, when the input is 0, there are 5 equilibrium points. One is this one, where both populations have low activity. So this is the resting state. So there is no stimulus. And this is where the trajectory is going to start. But there are also this input here, which is a saddle point. These are saddle points. And these two are attractors. Okay. And the saddle points, they have an unstable manifold that more or less looks, they have a stable manifold that more or less is like this. And they have a stable and an unstable manifold. Actually, the unstable manifold enters into the attractors here. But the important one is the stable one, because this one separates the basin of attraction of each of the 3 attractors in the system that we have. We have, remember, we have this attractor here. This attractor here, this attractor here, and this attractor here. And this stable manifold separates the region of attraction of the origin, which is this area here, the basin of attraction, from the basin of attraction of this other state here, okay, here, and the basin of attraction of the other attractor here, okay. But when we start here at the origin, at a low activity state, where both populations have low activity. And then the input is presented. And now let's assume that the coherence is 0, in the sense that both populations that receive the same input. So now we apply this input. Okay, so I1 equal to I2 and different than 0. And what happens is that now the null planes look like this. Now they intersect at 3 points. These two are attractors, and this one in the middle is a saddle point. And its stable manifold is like this. And we start here. We were at the low activity state, so we were here. Okay. What happens? That we are on the stable manifold. And actually, if the system was deterministic, basically this would tend to the saddle point. But the problem is that there is a little bit of noise. This is something maybe I didn't put much emphasis on the course, but the data and all the recordings and everything from experiments is noisy, all the signals, okay? So there is noise, there are effects of other neurons in the brain, other brain areas inside also the same population. This is a little bit noise. And the noise causes that in some trials, the trajectory is gonna, in some trials, the trajectory is gonna stick first to this stable manifold, but then due to the noise, it's gonna land on this attractor. But in other trials, what's gonna happen is that the trajectory is gonna stick to the stable manifold for a while, but then at some point, it will, due to the noise, it will land on the region of attraction, on the basin of attraction of the other attractor, and it will land on this point. And this point here, notice that the firing rate activity of population number 2 is high, while for population number 1 is low. So when we land on this attractor, we take decision number 2. And when we land on the other attractor, we notice that in this attractor, R1 is high and R2 is low. So we take the decision number 1. Okay. This is when the coherence is 0. So what happens is that, notice that we spend a lot of time along this, this stable manifold. So to the noise, to kind of like move the trajectory away from it and towards one or the other basin of attraction, okay, it takes longer. So the decision time or the reaction time to take a decision is going to be longer, okay? Let me comment another thing. In some experiments, instead of like letting the subject to take the decision whenever it's ready, they show the stimulus and they ask the subject to wait a little bit. So they remove the stimulus and then they ask the subject to wait a little bit before to take the decision. So the subject must kind of encode in the brain and remember that decision and hold it until it is asked to release it. So what happens is that once the stimulus is presented, the trajectory lands either in one or the other attractor. And then once it is removed, we are back to this phase plane. And what happens is that if we were here, now when we move back to the case where the stimulus is removed, we are here in this attractor. And actually we are holding this information until we are asked to release it. And this is how working memory, well, simplified working memory works, okay? So when we are asked to do tasks related to working memory, which is memorize or remember something for a short time, like when you're asked like, oh, remember this telephone number because I don't have paper now, so I'm gonna write it in 2 minutes, no? While I'm looking for the paper to write it down. So this task, which is working memory, so what's happening is that there are neurons in the brain that they are capable to stay at a high activity state, even when the stimulus is removed, okay? To keep the information about the stimulus. And then once you're asked to release this information, to retrieve this information, then the activity goes down. This is just a short parenthesis about working memory, okay? Sometimes this type of dynamics is represented by this kind of hypothetical energy functions. It's like when the stimulus is 0, you have this sort of energy function with 3 wells, and you are in the middle. Once the stimulus is presented, then what happens is that you land on this maximum. So you are on this maximum, and then the ball may go to the right or to the left. And basically, it's noise what's going to determine if you're going to land on the right or on the left. Okay. And actually, what's going to happen is that in 50% of the trials or the simulations, you're going to land on the right and 50% on the left, because in this case, there is no information from the external input. If now we consider the case in which there is a certain percentage of coherence, okay, like for instance, 6% of coherence, what it means is that the input, in this case, towards population 1 is a little bit higher than the input to population 2, okay? So this biases a little bit the Bayesian of attraction of the 2 attractors. Notice that now this separatrix, the stable manifold is not on the identical line, on the identity line. It's a little bit biased towards the right, in the sense that the region of attraction of this attractor here, where R1 has high activity and R2 has low activity, is a little bit bigger. So when we start here, okay, so when we start at the resting state, we start and we are inside the Bayesian of attraction of this attractor here. But what happens, so most of the trials are going to bring us here. However, due to noise, since we are in the Bayesian of attraction of the first attractor, but we are close to the separatrix, so we are close to the Bayesian of attraction of the other one, in some cases, the noise may bring us to the other side, and we are going to end up landing on the other attractor. So when the percentage of coherence is low, we are slightly biased towards one of the attractors, but only slightly, in the sense that more trials are going to end up on this attractor, but there are going to be also a lot of trials for which you are going to land on the other one. When we increase this coherence, notice that now the Bayesian of attraction of the first, so now I1 is much larger than I2. So this means that now the Bayesian of attraction for this one is much larger, and we start much more inside and far away from the boundary. So here the noise, it's much more difficult that it produces a jump in this other region of attraction. And here almost 100% of the trials are going to land on this attractor here. And we're going to take decision number 1. If this is 100%, the other attractor disappears. And now the null planes intersect only at one point, which is just R1 high and R2 low. Okay, so that's the dynamics behind this decision-making process. Since, well, I don't know if I have 2 minutes to just... Of
course, even 10 minutes.
Okay.
It's such
let me say that there are experiments in visual perception where the stimulus that is presented is ambiguous, in the sense that it admits different interpretations, and both interpretations are equally possible. Similar to the case in which, in the case where the coherence was 0%, in which like we had 2 options and both options are possible because it's random. So here something similar happens in the sense that we have a visual stimulus from which we don't have any information about like what is the correct percept, because all the percepts that we can generate out of them are correct. For instance, one famous one is the Necker cube, where you can see either this face as closer to you, or you can see the cube as is this other face is closer to you. Okay, so these are the possibilities. This other example is the face and base, where you can see either two faces or a base. And there are many, many figures in the literature. This is ambiguous figures, okay? And the two percepts are possible because it's not that one is correct and the other one is not. And what's happening in our brain when we see this? Well, when we see this in our brain, well, let me say here, that only one, so we only see one percept at the time. So we don't have a mixture of both in general. And they are sort of exclusive. But if we look at these figures for a long time, perception, we don't stick to one percept. Otherwise, perception changes over time. So now you see percept 1 for a while, and then percept 2 for a while. So switches are produced, okay? And these switches are random in the sense that it's not something periodic that you see one percept for 5 seconds and the other one for 5 seconds, etc., or something very regular. This is pretty random or stochastic. What is the mechanism behind? Well, the mechanism, so what's happening in the brain when we observe this is, again, that we see neurons whose activity is correlated to perception, in the sense that when the subject reports, I'm seeing percept number 1, there are a certain group of neurons that are active. And this group of neurons are suppressed or have low activity when the subject says, I'm perceiving the other percept. So we have neurons in the brain that are correlated to perceptions. And how do we model this? And the model is similar to the ones I showed you, in the sense that we have two populations that they inhibit each other because they compete, so that when one is active, it suppresses the other one. Okay, it sends inhibition to the other one. And they may have self-excitation as well. But in this kind of processes, we add an extra term, which is the adaptation. And this is a slow process, a slow negative feedback, in the sense that the neurons, after a while, they fatigue of spiking. They get tired of spiking. And what happens is that those neurons that are active, that because we are seeing percept number 1, after a while, the adaptation produces that its firing rate decreases because they fatigue of firing. And this causes the other neurons to activate and provide the switch. This mechanism, together with noise, produces these random switches between percepts, okay? So similar to this idea of like these hypothetical energy functions, this is the situation that we've seen with with decision-making, right? That we are here and we may land in one of the attractors, and when we land in one of the attractors, it means that we are perceiving number 1. But adaptation, what it does, it sort of like smooth the depth of these wells, or maybe it makes them disappear. Okay, so that then the trajectory moves to the other well. Okay, and this produces switching. This is with just noise. It could be just with adaptation. It could be only with noise that produces these switches, or it can be a mixture of both. And actually, most of the models go for this mixture. And now I'm going to the end. Just closing remarks, because this is the last lecture of the course. So, this field is very broad, and for every lecture, you could like find like many papers talking about these topics. Okay, so there is a large literature. So this is just a showroom of some classical models, or like some well-established models in the field. But of course, there are many models, many studies, many areas, many diseases, many cognitive styles that are studied. So it's a very broad field. And actually, you can see that I mainly focused on dynamical systems, and late in this late part, like I mentioned, that there is noise, so they are stochastic differential equations. But of course, there are many mathematical techniques that can be used to understand, to tackle these problems. And the number of open questions is huge, because the brain is still an unknown. But so in this last lecture, I tried to show you a little bit about like decisions. You can see how complex it is to set an experimental, to put an experimental setting for decision. Or there are many, another big field or big area of research is like memory, long-term memories, short-term memories, working memory, etc. Also like how information is encoded, how communication between areas. There's also this topic of oscillations that I didn't discuss much here, but oscillation, synchronization, phase lockings. So this is another big area of research. And it's been a pleasure. And here you have my email in case you want to request information. That's it.
Thank you very much for the wonderful lecture and the wonderful mini course. I think we should clap. People can open the microphone. So there is a raised hand. Someone raise the hand. Can talk, please. I think it's, I see the raised hand.
It's who I see here is Serginei.
Yes, please. You can talk. Maybe his internet. There is a question from YouTube from Michael. He is asking previously, when you were talking about mean field, if when you talk about mean field equation, you're talking about ODE or PDEs.
So yeah, you need to write. So this mean field derivation is based on like writing a PDE, which is the Fokker-Planck equation, that describes this mean firing rate. Okay. But then the problem is like how to solve this PDE. And then, and this links to the previous talk on Tuesday. So sometimes there are like some assumptions that allow to move from this PDE to an ODE system. And actually, what I discussed today was ODEs. So this mean field are ODEs. But there are some hypotheses that need to be assumed, and the reduction in the end is not an exact description of the firing of the spiking network, in the sense that if you simulate the spiking network and you compute these averages, you're not going to obtain the same quantitative value as if you simulate the reduced model. But lately, in the last 5 years, there's been several studies and several results on mean field models that describe exactly the firing rate of the spiking network. Of course, some assumptions need to be taken on the type of cells in the network, etc. But at least you have that. If you simulate the spiking network with a large number of neurons and you simulate the reduced model, you obtain an agreement, a total agreement in the evolution of the variable. But this mean field approach, so I talk a lot and I don't know if I answered your question, but these mean field approaches involve the Fokker-Planck equation, which is a PDE. Although you try to so finally you obtain an ODE system, which is the one you study as the reduced model.
Yes, there is another question.
Could you list some names of labs which do research experimentally and dynamical systems model? Well, there are several, and there are many, but so I'm going to mention the one from which I learned while I was doing a postdoc there. So I was in the Center for Neural Science at NYU, and the Center for Neural Science collaborates with scientists or with mathematicians at the Curran Institute, but also with other in the physics department, with other people doing modeling. And it also has labs in the center that are like computational labs. So most of the labs are experimental, but they also have computational labs, and there are a lot of interactions between these labs. But in Colombia, there's also people, but there's people, it's que there are many.
Yeah. There are thanking you even from YouTube for the previous answer.
Okay.
So, more question. I had two very basic questions, because first, when you showed the two equation models, it's only the firing that you're modeling. So the neuron can receive, is receiving and the fire. So it's the receiving part. How much it can receive is not model. It doesn't enter in the model, because it either receive or it's firing, or the two activity can be happening at the same time.
So the activity, so you mean this equation here?
Yes, yes. Can he fire and receive at the same time? Can he have this, or this is a sequence? First he fire and then he receive. Is this receive is at the same time is firing in gene and the receptor, or these are activities that have to happen in sequence?
So the way it is modeled here, it's instantaneous. It happens at the same time, in the sense that as for instance here, as if R1 changes, it is immediately felt by population R2, immediately. There are models, you can make variations of this model and make that this that the effect of the changes in the firing rate activity of the population are felt with a certain delay. You could put, like, for instance, a variable here, S1, and that S1 has its own dynamics. I'm gonna do the most easy one, saying that it's not immediate that when R1 changes, it is immediately felt by this. It goes through a process where S needs to build up and with a certain time constant before it can affect this population here. But the way this model is designed, it's very simplified, because everything is immediate. And this is a little bit, so here there is a little trick that I didn't mention, is that there is a principle, the Dale's principle, that says that if a neuron is inhibitory, it can only, so being inhibitory or excitatory is a property of the neuron, in the sense that if a neuron is inhibitory, all the connections that this neuron is going to have with other neurons are going to be inhibitory. It is not possible that this neuron inhibits a neuron and excites another one. Okay, and here what we are building is populations So we are cheating because population 1 is exciting itself and inhibiting the other one. But here is because we did this trick or this simplification is that, no, inhibition, this population is excitatory. It excites an inhibitory population that at its turn suppresses the other one. But we are assuming that this is so fast that we can directly, mathematically, we can write down, directly write it like this. Although it's cheating. But mathematically, the output that we get or the dynamics that we get from this system is not different than if we had considered this intermediate neuron, this inhibitory neuron. And actually, this allows us simplification that is very useful then when we want to study the model.
And the other question I had when you showed that figures that you say that you can see a face or a vase. And then, yes, and then the fact that maybe, let's say we saw two faces, but then could an emotional state change this perception? So at a certain point, can we, because of something, see the vase? Because, you know, we have this common sense, be open, or these open mind eyes. And it does happen that something that we don't usually see, we see. You know, it can be an emotion, it can be visual, it can be things. So these perceptions, how this is triggered, you know, this sometimes it's really vision. It's really vision, you know. So how this is triggered, this change?
This is a good question, and this relates to the fact that it's hard to isolate these problems or to isolate these tasks from other outputs and other inputs that we are getting. So, for instance, in these experiments, it also blinking, it seems that it has an effect. It's like when we blink, so we tend to switch when they are blinking, eye blinking, or like so when we want to study these in the experimental labs, what they have to do is like ask the subject to sit in front of the screen, fixate the eye. So they put a fixating point, and they ask the subject not to move the eyes out of this fixating point, because you want to have as less other inputs as possible. And also they have to be careful, like taking like breaks so that the subject doesn't get tired. Because of course, our reports of like what we see are gonna be affected if we get tired of looking at a figure. So they have to take breaks. They have to be careful. So it's tough to do these experiments because you have to be careful about many details that may distract you or may affect the output. And of course, there's a lot of variability in subjects. There are subjects, when you do these experiments, there are subjects that have very rapid changes and subjects that they have like very slow changes, in the sense that they stick to a percept for much longer time than others. And also they say that people that they do meditation, they can stick to one percept for longer time than other people. So that's another problem in neuroscience, not only for these experiments, is that there is a lot of subject variability. So you try to set the experiment, so to set like all the boundaries of the experiment and everything, but subjects, we are very different, and it's hard.
Thank you. It's very fascinating, all this. As you say, there are many.
And it's complicated, no? Because sometimes we as mathematicians, we want to find general rules.
But this is why I appreciate this the Dutch, that they stress the individual.
Yeah.
You know, I think it is the way to go, because They try to map the circuit of everyone because I think that we are all different. We are all different for the best, for the worst, you know. I think it's good that we are different, but.
But actually, as you can see in this model, so there are many parameters in the model. So I didn't put much stress, but you need to decide how strong are the connections. And this is a parameter that you can vary, but the inhibitory connections, the excitatory ones, so the external input, all these things are parameters that are going to produce like outputs that they're going to have like people who are faster at taking decisions, they're going to produce people who are slower at taking decisions, etc. So even when you find a model, you can put variability in the model, just modifying parameters.
Great. So I think there are no more questions. I'm just checking 1 second once more. Now they say thank you for a nice talk. Yes, there's one.
There is one on chat.
Yeah.
Yes.
I have a question regarding the previously mentioned model with R1 and R2. Theoretically, the models predict that if the intensity of external input for 1 and 2 are the same, the subject can fall in a no decision zone on the null line. Can this situation of no decision happen in real life? The task is called two-choice forced task because the subject is forced to take a decision. If not, so in real life, sometimes there's people that they cannot take decisions because they don't see. Since when you don't see what is the good decision, or like you don't see the decision like clearly, some people cannot take the decision, even if, unless they are forced to. And you may think that when there is this forcing, also there is something that produces you that you have to. It's either way, you go to one attractor or the other, because actually the option of staying on the stable manifold of the saddle, I mean, this with a little bit of perturbation, it's this is totally unstable in the sense that either you fall on the, the option of getting stuck here is not an option, in the sense that a small perturbation would bring you either to the right or to the left, and then you're going to land on the basin of attraction. What happens is that you stay longer, so it takes longer to take the decision in this case. But at some point, you're going to land on one of the attractors, because this is unstable. And in real life or in biological systems, actually, unstable objects are only studied because they help us to understand how they organize the dynamics. But these are objects that we never land on. We don't see them in the experiments. We mathematically study them because they help to understand the dynamics and organize how the dynamics of the system is organized. But in the end, these are points where so biologically, we don't see unstable fixed points. So the trajectories are never an unstable fixed point. Unless it is, let's say, very weakly unstable in the sense that it takes a long time to move away from it. Like right after a Hopf bifurcation, we can observe this, that we are on an unstable equilibrium point. And since the rate of repulsion is very weak, so it takes long time to move away. And you can see that for a period of time, you're spiraling around and moving away. But you don't stay on unstable objects.
And speaking about invariant manifold, the next mini course is Angel Jorba. So you will hear a lot about stable and unstable manifold with other applications, but the math, there is some similar math you can use. So thank you to everyone. Thank you to the students also, because, the questions are very important. So thank you to everyone, and to the one in Europe that is very late. Thank you for staying with us.
Thank you.
Rodrigo is even ahead of everyone, I guess. So thank you once more, Gemma. Very nice,
you.
beautiful course. And we hope to see you in the future with us. And all the articles are very welcome. And again, for everyone, next course will be in June. And we'll have another person from the Barcelona group. So this group that is so much important for us for all this beautiful application. So we are happy to be connected to the Barcelona group in this remote way, you know. Thank you very much to everyone. So see you in June. Okay. Thank you, Gemma.
Thank you.
Thank you, Rodrigo.
Thank you.
It was
Thank you.
wonderful.
Yeah.
We'll
Thank
see
you.
you all in June. Well, hope that we could see you all in Barcelona.
It would be nice to see in Barcelona.
Yeah, Barcelona is a very nice city.
Okay. Bye-bye.
Bye.
Bye-bye.
Bye-bye.
Good evening for the Europeans and
Thank
good
you.
afternoon.